and thank you for tuning into this presentation at the AWS HCLS Virtual Symposium. My name is Arne Materna, and I'm the CEO of ARESdbaleading. Over the course of the next 15 minutes, I will speak about how machine learning and smart bioinformatics can overcome key limitations in genomics-based infectious disease testing. ARESdbaleading is a Vienna-based startup founded in 2017, and we are, together with Predictive, part of the OpGen Group of companies. Together, we fight against antimicrobial resistance through cutting-edge molecular microbiology, with solutions for the rapid diagnosis of severe infections and for infection control. While Predictive and OpGen bring molecular diagnostic assays to clinicians and patients, ARESdbaleading is combining next-generation sequencing with smart bioinformatics and artificial intelligence to address the global threat of antimicrobial resistance. Let's begin with a few facts on AMR, or antimicrobial resistance. While the attention of many of us is quite rightfully on the impact of the current COVID-19 pandemic, another severe pandemic is on the rise. Antibiotics, which we all have come to rely upon as effective treatment of otherwise often dangerous, sometimes deadly infections, are slowly but steadily becoming ineffective. We see a global increase in pathogens that prove resistant against standard and even last resort antimicrobial therapy. With already tens of millions affected by AMR and dozens of billion euros per year in direct healthcare costs, the most severe consequence we face are over 700,000 fatalities each year. That is over 2,000 fatalities per day. These numbers are expected to rise, and some reports calculate these numbers to reach 10 million per year by 2050, meaning AMR would eclipse cancer as the leading cause of death. The problem of AMR is exacerbated by the challenge of choosing the right treatment. Predicting how an infected patient will respond to antibiotic therapy is a hard problem, influenced by factors such as the genomics of the pathogen that is causing an infection. As a consequence, only a minority of patients are receiving appropriate treatment, a fact that has become apparent once more in the overtreatment of COVID-19 patients with antibiotics designed to quell bacterial co-infections. In case you're wondering why overtreatment is a problem, it is worthwhile to take a look into what is actually causing antimicrobial resistance, or AMR. As any microbiologist will explain you, antimicrobial compounds, or antibiotics, are produced by microbes themselves in order to compete with other microbes inhabiting the same ecological niche. In response to these compounds, microbes evolve the very mechanisms that cause resistance and that give us trouble in the care of infectious disease patients. Inappropriate antibiotic use, for example, overuse or simply using the wrong antibiotic for a pathogen, then selectively enrich for microbes that have evolved such resistance mechanisms. And whenever this situation is met with imperfect hygiene or imperfect infection prevention, then we face the risk of spreading antibiotic or multi-drug resistant pathogens. And that in turn requires increased antibiotic use, fueling this vicious cycle. The question therefore is, what can be done to break this cycle leading to AMR? On one hand, we rely on the pharmaceutical industry to keep developing novel forms of antibiotic therapy. The challenge for pharma thereby is that new drug development becomes a race against the evolution of resistance mechanisms. This, of course, is a very tall order. While an encouraging number of 70 drugs is in different stages of clinical and preclinical development at the moment, the WHO also notes in the just released 2020 report on antibiotic drug candidates that this may not be enough to tackle AMR, as currently over 80% of newly approved drugs can be rendered ineffective by existing resistance mechanisms. This does, however, by no means mean defeat. Improved antimicrobial stewardship can help us regain the advantage over AMR. Antimicrobial stewardship refers to technologies and interventions that promote the optimal use and avoid the overuse of antibiotics. Now, why is this important? As I just alluded to when addressing the cause of AMR, the appropriate use of antibiotics was shown not only to improve patient outcomes, but also to reduce the emergence of antibiotic resistance. And this is where technology can help us make a difference. In order to promote appropriate antibiotic therapy and enable antibiotic stewardship, we need solutions that diagnose or predict resistance, to enable a targeted therapy with appropriate antibiotics. And we need solutions that enable global surveillance for AMR to allow us to detect outbreaks and counter them with infection prevention measures. Let's take a look at the technologies that are in the clinic today. The standard of care in infectious disease diagnosis are bacterial cultures, which are routinely used for the identification of infective agents and for the determination of suitable antibiotic treatment options. When a culture is positive, meaning if pathogen growth is present, then antibiotic susceptibility testing, also referred to as AST, is performed in order to determine which antibiotics are effective and which will likely be met with resistance. While these culture-based methods are being considered the gold standard, they are often labor-intensive and they may miss difficult to culture pathogens. Also, turnaround times range from 1 to 3 days or even more. And this is long considering that patients with acute infections require urgent care within hours. This lack of sensitivity and speed is addressed by the more modern molecular tests relying on PCR. Leading PCR panels, such as the tests available on the Curators Univero system, achieve very high sensitivities and can return results in a matter of hours, even pushing the limit below a single hour. However, these systems can only screen for up to a few hundred biomarkers at best. This means each assay, which has to be individually developed, validated, and potentially approved, can cover a limited number of pathogens, and for each pathogen, only a limited number of genetic markers for antimicrobial resistance. Given that today the number of known genetic markers for AMR is measured in the thousands, the question has to be, is there a system that could simultaneously and comprehensively screen for any pathogen and all known genetic markers for antibiotic resistance? And what if the system could not only report the detected AMR markers, but also emulate the advantages of culture by predicting whether detected pathogens are susceptible or resistant to antibiotic therapy? And here is where next-generation sequencing, or abbreviated NGS, enters the picture. NGS is increasingly adopted by clinical labs, and its utility for microbial typing in epidemiology is already undisputed. It is the first technology that, thanks to an enormous sampling depth, is not only very sensitive, but also captures the entire genome, allowing for an exhaustive characterization of genetic AMR markers in a single test. More comprehensive genomic information on pathogens then, in turn, provides the chance to use artificial intelligence and predictive modeling to emulate culture-based antimicrobial susceptibility testing and to predict which antibiotics represent suitable treatment options. With sequencing costs and turnaround times rapidly decreasing, trying to overcome the shortcomings in the current standard of care through sequencing-based predictive antimicrobial susceptibility testing has firmly emerged as a focus for ARESdbaleading. Allow me to share with you some of the key challenges, but also milestones we've met on our mission to revolutionize infectious disease testing. Once our objective had been set on enabling accurate and fast detection of pathogens and the prediction of AMR, we started to address potential roadblocks. Very quickly, databases for AMR markers emerged as a critical technological component. When we evaluated several public AMR marker databases for their suitability for predictive AST, we recognized several shortcomings. Here are 2 examples. The annotation of AMR markers at the level of antibiotic classes was too coarse for machine learning, considering that different antibiotic compounds in the same class are affected differently by any given AMR marker, for example, because of different enzymatic affinities. Therefore, raising the resolution of marker annotation from antibiotic class to the compound level substantially improved predictive modeling. Moreover, simple rule-based models for individual markers, they tended to overgeneralize, because multiple genetic factors can, for example, synergistically or antagonistically contribute to the antimicrobial resistance or AMR phenotype. We needed to adjust for biology and allow for polyvariate predictive models. Based on these and other findings, we decided to develop ARESdb, a comprehensive AMR knowledge base designed from ground up to enable predictive antimicrobial susceptibility testing. ARESdb today is a globally leading database of curated AMR markers and information about resistance derived from genotypic and phenotypic data. It harbors a continuously growing collection of clinical pathogen genomes and associated, validated antimicrobial susceptibility test measurements that determine how a given pathogen in this database has responded phenotypically in the lab when exposed to a wide range of antibiotics. And finally, ARESdb covers a number of predictive models that we train based on these genomes and the associated resistance phenotypes. Currently, the models to predict antibiotic susceptibility are trained as stacked ensemble models on AST results and on a variety of genetic markers, including k-mers and gene protein annotations within the ARESdb. This model-based approach can be complemented by a rule-based approach, taking into account the interplay between multiple resistance mechanisms and antibiotic compounds. As the figure in the middle of this slide illustrates, we not only rely on the presence-absence of certain resistance genes, but we also consider changes in non-coding regions of the genome, as well as novel markers that our predictive algorithms have discovered. We demonstrated in several studies the high categorical agreement between standard of care results with the resistance phenotype predictions we generated based on NGS data. These studies include, for example, the first and only multicentric evaluation successfully demonstrating the feasibility and the performance of predictive AST for over 13 pathogens. To help translate this technology into application, we have recently validated the entire end-to-end workflow, from sequencing on Illumina, representing the de facto standard NGS platform, all the way to the data interpretation via ARESdb and our AI-powered bioinformatics. We did apply CLIA standards to the validation process and have published both the performance as well as the validation protocols to make ARES technology portable to any other NGS laboratory. The ARES solution spectrum today includes a number of next-generation sequencing services as well as software. Central to our offering is and remains the proprietary ARESdb. This database both powers ARESiss, our service for bacterial isolate sequencing, as well as the ARESupa. ARESupa is a universal target enrichment NGS panel that comprehensively covers pathogens as well as thousands of genetic markers we've selected from the ARESdb. And it is currently used in collaboration with 5 early access customers. And finally, ARES Cloud is a cloud-based user-friendly interpretation and reporting software, which enables the ARES NGS lab and will be later this year made available to other laboratories. None of this, of course, would have been possible without the trust and support from our partners, which include, for instance, Sando, QIAGEN, the Austrian Public Health Agency, AGES, and a number of clinical collaborators in the EU and the US, including Johns Hopkins Medicine. Researchers at Johns Hopkins have, for instance, assessed the suitability of new nanopore sequencing technologies for predictive AST, and were able to demonstrate a predictive performance for this new data type that's coming very close to the performance of Illumina data. Another story I'd like to share was a research collaboration with Sando, a global leader in antibiotic development. The rising prevalence of multi-drug resistant pathogens is limiting the use of modern last resort antibiotics. In order to better protect these treatment options and reserve them for severe cases of AMR, Sando investigated in a proof of concept study with ARES if ARESdb and AI could be used to predict if also older antibiotics, when effectively combined, can be used to circumvent AMR. Our predictions were that the use of fosfomycin and amikacin would outperform modern treatment options, including piptazo or meropenem. And importantly, these predictions could be experimentally validated in this pilot with a promising categorical agreement of 85%, certainly warranting further follow-up. Finally, the partnership between ARES Genetics and QIAGEN demonstrates that in-licensing of the ARESdb contents can help other bioinformatics providers to overcome key limitations of public AMR databases, such as the lack of diagnostic performance information that's assigned to individual AMR markers, for instance. Last but certainly not least, I'd like to conclude by thanking our infrastructure partner, AWS. not only for the opportunity to present here, but AWS truly is crucial to R&D at ARESdbaleading. ARESdb curation and AI model training rely both heavily on on-demand scaling, which has proved essential for our training sprints. We optimized our training algorithms and orchestration systems using AWS Batch in the background, for example. This allows us to quickly retrain all our models based on our reference database at cost. For early stage exploration as well as routine updates, that's a significant advantage. Ares Cloud is of course also developed and deployed on AWS using various serverless and related services. Doing so results for us in a lower burden for software development and agile DevOps while maintaining high security standards. Our data processing pipelines can run in a fault-tolerant fashion at relatively low cost, making commercial software development economically more feasible for us. And with that, I'd like to conclude and thank you for your attention. Please don't hesitate to reach out with any questions that you may have. Thank you very much.